This is an example of, I'm not going to, I can't show you every single paper published in the last 20 years. This is an example, also because this paper has nice graphs, of the paper by Viswas et al., in which they're looking at VRE in mice. So you can see here that groups of 5 mice were challenged with VRE, and then treated with the various doses of phage at 45 minutes after challenge. And they were then followed out, to 6 days. So you can see that there's a dose dependency with phage. Or of course, if they had a dose of no phage, their health was 0, which is dead. And so 5 means healthy. This is based on clinical science. So as you can see, the higher doses, all the mice survived when given higher doses of phage. They're both IP in this one. So and I'll talk about this a bit about how they tend to do the treatments of these. So they're both IP injections in mice. So you can see that there's a certain dose response effect, and this is a treatment time effect. So from the same paper. So if you look at the mice were all challenged, and then they, instead of treating them all 45 minutes, they treated them at 5, 18, and 24 hours, and then followed them for 20 days to see how well, it is very important to treat them immediately after infection. So you can see you still have some survivors out here, the ones which are treated. If you challenge them with the bacteria and then wait 24 hours, you do have 2 survivors out of 5, but the difference here is you have they're totally healthy and then 3 that are totally dead is, if you wonder what happened with those 2 mice, they're kind of like outliers, I guess. But you can see there is certainly an effect of early treatment. If you wait too long after challenge with the bacteria, the treatment becomes less effective. So this is a pretty important experiment that was done by Carl Merrill. Carl Merrill's other big claim to fame is he developed silver stain back in the early 70s for proteins. So he did this experiment to look to see if you could get phages to circulate for a long period in mice. And it's a very nice and elegant experiment. So in this case, he used just a lytic mutant of lambda. So it was lambda phage, which is a tempered phage, but in this case, it's unable to form lysogens. So it will form clear plaques, and it works as a virulent phage in the context of this experiment. So if you can see in this line right here, if you inject the wild-type phage into mice, so this is intravenous into mice, and then you take samples out over time, the number of phage remaining goes down quite quickly, about 5 logs in a day. So what he did was he would take this wild-type phage, passage it through the mice, and wait for only 7 hours, and then take a sample out, take those survivors, and then grow them up again, and then re-inject those into the mice, and then wait another 7 hours, take those out, and grow them up again, and repeat that 10 times. So you have actually, it's a very nice and I think very elegant population selection experiment. So you're not doing experiments say where you're doing a selection, you pick off a single individual. You're applying your selection pressure, which is to put them into the mice and into the circulatory system, and then you're just removing the population that survives. So your expectation is that the population of these will be enriched in mutants, which are able to circulate for longer, hopefully. So he did it for 10 times, although actually in the paper he says after the first 4 times, he pretty much had, they were circulating pretty much the same as the ones that had been passed for 10 times. So he kept 2 phages, he called Argo 1 and Argo 2, the long voyage. And you can see over the same time period compared to wild-type phage, they dropped only about a little under a log unit in the same time period. So these phages are able to circulate for much longer in the mice compared to the wild-type. And then he just did an experiment with an experimental bacteremia model in the mice, which is a bloodstream infection. And so the phage in the bloodstream. And so you can see here, this is your no phage control, and this is the disease symptoms here. So 5 is not good in this case. So it's the opposite of the previous one. And you can see the long circulating phage. I know I think I have it backwards. So the long circulating phage work better in this model. Sorry. Oops. However, he did some proteomic work on the long circulating phage and found that both of them had the same mutation, which is a single change from a lysine to glutamic acid in a major capsid protein. So that single amino acid change in major capsid protein allows them to survive in the host for much longer. So now let's talk a little bit about. Sorry.
Before we get so they should have survived longer, like say over a day.
Yeah.
Did they have any data showing, if you come back a week later, they still be exactly that?
So the previous graph showed that these long circulating phages could survive, on a day scale better than the wild type. Are there any data on just how long they stay in the bloodstream, like at detectable levels?
Yeah, that is mentioned as a data not shown in the paper, and they do mention that they can detect them out to several days. Of course, the titer is going down to it's like 10 to 3 per mil at that point. Yeah, but it does go down. It goes down actually, pretty linearly from this line. It just if you just follow that out, say to 5 days, you can still get some phage out.
Do you know how the mechanism for clearance?
Well, it's clearance by the reticuloendothelial system, which pretty much means they tend to all end up in the spleen. So they're just filtered out of the blood over time. And so this is not an acquired immunity thing. This is an innate immunity. Now, of course, if you continue to challenge them over and over again, you would expect to see acquired immunity show eventually because they are antigenic, and you would start getting antibodies produced against them. So this is some work, some animal work, this is actually a picture I took during my PhD work when we did animal trials on dairy cattle. So an attractive target for doing phage therapy work is animal systems, because number one, you can actually treat the actual clinical syndrome that you want to treat. You're not worrying about a mouse model of a human infection. This is the actual infection that you are interested in the actual animal that you're interested in treating it. And number two, the barrier, the regulatory barrier for treating for veterinary use is much lower than it is for human use. So you can go into, you don't need to do a whole bunch of phase 1 and 2 clinical trials if you want to go do experimental treatments in animals, both animals. So there's been a bit of work, a few papers coming out on treatment of 01578 in cattle to reduce shedding. There's a few groups that have done this. We call it 01578 is something called the hamburger strain, with the large jack-in-the-box outbreak in the 80s. It's still considered one of the major problems in food safety. So it spreads among cattle in crowded feedlot conditions, and the cattle carry this asymptomatically. They don't carry the receptor that the Shiga toxin binds to, so they just carry it in their colon, and they don't mind. However, once they go into slaughter and the carcass is cut open, then you can have contamination of the carcass with the 01578 that was part of the intestinal contents. And then it makes its way into the meat, and then particularly in hamburger, where it's all ground into the meat, it can be a major problem. And there was a recall not long ago of ground beef due to 01578 contamination. It's very dangerous. The infectious dose of E. coli 015.7 is about 10 cells. So the regulatory limit for 015.7 actually is 0. If you detect a single cell of 015.7 in your sample, the lot has to be recalled, and that's the regulation. So there's a few groups that have shown some efficacy of up to a few logs in reducing 157 carriage in cattle. Some groups have done it just by straight feeding. There is a company which has developed a product which is a microencapsulated phage, which will go through the rumen and makes its way through the rumen and into the intestinal tract that way. So these they appear to be effective, so that this is also an attractive target for companies. So there are a couple of companies which are apparently rolling out products at some point, although once things go into a kind of a company pipeline,